Imagine if we could cure Parkinson's disease, or Alzheimer's, or blindness, or be able to enable someone with paralysis to walk again, or repair a damaged heart, or perhaps grow a new one for you. On Monday, our newly elected president is about to remove some of the restrictions on embryonic stem cell research, which claims to be able to offer us medical treatments that will be able to make these dreams a reality. Understanding stem cell research is going to be important for you because you're going to hear it in the news. People are going to be discussing the issues, and being able to understand some of the terminology, some of the issues and controversies that are involved will help you to be able to engage in those discussions. So first, I'm going to tell you what stem cells are and how they work, and I'm going to share with you exactly what embryonic stem cells are and how we get them and what they do. And I'm also going to share with you some of the recent developments and what we can do with them today. So in short, a stem cell basically is a basic building block for human life. Every cell that you have in the adult human body actually came from a stem cell. And so the stem cell has the potential of becoming other cells, and this is what makes them so important. There are basically three different kinds of stem cells. There are more, but I just want to bring out the three for you. One is embryonic stem cells, those which we abstract from human embryos. Those are said to be pluripotent, meaning they have the greatest potential of becoming any cell in the human body. Also, there is umbilical cord stem cells, which we can use the blood in order to utilize those stem cells, but they don't have as much flexibility to them. And also adult stem cells. The only stem cells in the adult human body are adult stem cells, and they usually serve the purpose of repairing. And so they have some flexibility, but not so much as the embryonic stem cells. What makes stem cells important, they usually have two characteristics. One, they can go through ongoing mitosis, meaning that cell division is continuous. In other words, if you have some stem cells, you could have them reproduce or replicate themselves indefinitely, to have an unlimited amount of stem cells. And second, they can become differentiated, or they can become more specialized to serve different functions within the body. And so it is the task of a scientist to be able to recreate differentiation by external and internal cues. External cues by means of chemicals, or by internal cues meaning genes, that we can somehow replicate exactly what happens in the human body in order for us to be able to make better use of stem cells. I want to talk to you a little bit more about the embryonic stem cells, because those are the ones that we hear about in the news, and those are the ones that are more controversial. Embryonic stem cells basically come from what is called a blastocyst, and this is a part of the embryonic development, somewhere between the two gametes forming a zygote until 8 weeks, when we were considered to have a fetus in human development. This blastocyst is the key point. Reason being is because this is before implantation occurs. This is before 5 to 7 days. And at this point, we can go in and remove what is called the inner cell mass that has these stem cells. And by doing so, we destroy the embryo. But these stem cells that we get are the most flexible, and the ones that scientists like the most, because they have the most potential of becoming any cell in the human body. There are a few different ways that we can actually get these stem cells. One is the most common, is in vitro fertilization. That is basically taking an egg and sperm and forming a zygote within a test tube. Or also leftover zygotes, human embryos, from fertility clinics. And these clinics usually have 5 to 7 extra fertilized eggs in case implantation doesn't occur in the first or 2 tries. And so that's the most popular. Another way is from aborted babies. Since the babies are freshly aborted, they could be donated to science and their stem cells can be used. The most interesting of them is called somatic cell nuclear transfer. It's basically how we clone. And any time that an egg is fertilized, it's normally the egg of the female and the sperm of the man, which brings the 2 halves of the 23 pairs of chromosomes. However, in this, what we do is we take the egg, we strip out the nucleus, we take the nucleus of a somatic cell, that is an adult cell that has the 23 pairs of chromosomes, and we put it into that egg so we can begin moving it into a blastocyst so we can remove the inner cell mass and be able to create a culture of embryonic stem cells in a petri dish. Third, just want to briefly sketch for you some of the recent advances that have occurred. It was 10 years ago that a gentleman by the name of James Thompson was the first to be able to remove the inner cell mass from a blastocyst, and so that we could be able to successfully remove the stem cells and be able to grow them in a petri dish. Within the next 3 to 4 years, this continued, and scientists wanted government funding. And President Bush, in August of 2001, said that we're only going to fund those stem cell lines, that is, stem cells that have grown over more than 6+ months and continue today. And it is important that these stem cell lines were going to be funded by our tax dollars. And then more recently, August 2006, Robert Lanza has claims to have successfully removed the inner cell mass without destroying, or removing stem cells without destroying the embryo. So that is one development for the concerns of those who have ethical issues with the embryonic stem cells. Also, with the use of viruses, they've been able to take adult skin cells and be able to make them into what appear to be embryonic stem cells. They look and act just like them. And most recently, within the last few months, the FDA has recently approved the first human testing involving embryonic stem cells. So this is groundbreaking. As well as tomorrow, President Obama is looking to restore scientific integrity by an executive order to lift the restrictions and be able to allow embryonic stem cell research of new embryos that will be destroyed to be funded by your tax dollars. So basic cures, up to date, no approval has been given to any medical treatments derived from embryonic stem cells. All of it has been derived from adult stem cells or cord blood stem cells. And so all of the benefits we have seen so far from this research is from non-embryonic stem cells. And in fact, we've been able to cure a number of blood and immune-related genetic diseases, cancers, disorders, juvenile diabetes, Parkinson's. We've cured blindness by this, and spinal cord injuries. And finally, Father Tad Pachalkic, Director of Education of National Catholic Bioethics Center, adds that adult stem cells, on the other hand, have already cured thousands. There is the example of the use of bone marrow cells from the hip bone to repair scar tissue on the heart after heart attacks. Research using adult stem cells is 20 to 30 years ahead of embryonic stem cells and holds greater promise. This is in part because stem cells are part of the natural repair mechanisms of the adult body, while embryonic stem cells do not belong in the adult body, where they are likely to form tumors or they can be rejected as foreign tissue. So in conclusion, understanding some of these basic issues, modern advances, as well as some of the terminology and controversies about it, should be able to do a quick view and enable you to deal with the issues concerning stem cell research. Okay? Stem cell research has come a long way in the last 10 years. And so with the changes with recent funding, there's going to be more talk in the news and perhaps in the workrooms. Thank you.